the PBMC Humanized mouse platform that we're using for a lot of different therapeutics, but my presentation today is on CAR T, showing efficacy, cytokine release, and expansion for autologous and allogeneic platforms. I'm going to give about 5 slides of introduction, then I'm going to jump into the data. There's a lot of slide data, a lot of data. So anyway, hopefully have time for questions at the end. Our lawyers want to make sure everybody understands that we filed some IP on the platform. Okay. About 5 years ago, when we first got involved in this platform, I went to a CAR T meeting, and I talked with 8 individuals after their talks about the issues of safety when it came to CAR T therapy. And we were seeing toxicities in my laboratory with bispecifics, and we're curious about what was going on in the case of CAR T. And the feedback I got from the people I talked to was very helpful. But one gentleman actually laughed in my face when I talked to him about the in vitro assay in correlation with what they're seeing in the clinic. And clearly, I get the impression that people were looking for something better, something different when it comes to understanding the safety profile of CAR T therapies. So anyway, we built this platform. It's not the normal PBMC platform you see in the literature. It's a new type of way for us to do it. But essentially, we've developed this platform for therapeutics that's fast, sensitive, reliable, and reproducible in vivo model for cytokine release syndrome to help de-risk antibody and cellular therapies for preclinical development. This platform is useful for us to look at high affinity, low affinity antibodies for the same target, looking at efficacy and safety, evaluate individual responses. The key here is we wanted to have something that was going to be able to capture a population's responses, not just a mouse response. How do individuals respond to the same therapy? I'll talk more about that in a few minutes. We did drug combinations, looking at bispecifics and antibody therapies in combination with each other. Efficacy and safety, and of course for CAR T, we're looking at efficacy, CRS, and CAR T expansion. The platform is robust. Depends on the mouse model you use. I have access to about 24 NSG variants. And most of the data I'm going to show you today is with the mouse MHC class 1, class 2 knockout mouse, which is basically an absence of acute GVHD or a delayed GVHD. I can tell you more details about that later on. It's not a perfect model, but it's better than other strains out there when it comes to the onset of GVHD. But essentially, our platform, we can capture T cells, B cells, NK cells. We're getting monocytes and myeloid-specific cytokines and cell populations in various organs as well. So we're capturing a very robust human immune system with PBMC engraftment. And then for human cytokine induction, obviously the T cell mediated cytokines, but also myeloid panels, and some of the cytokines we show here are some of the ones we see. Different cytokines are induced by different therapeutics, and antibody therapy versus a bispecific versus a CAR T. You'll see different patterns of cytokine induction, which shouldn't surprise anybody. Our approach is a holistic approach, looking at essentially as much as we can within an individual mouse. We only use 5 mice per group. We don't need to use more than that. We get pretty tight data in our platform. But here is we look at cytokine release syndrome in relation to efficacy. We look at cytokine induction, body temperature changes, mouse clinical evaluation. How does the mouse look essentially? Downstream organ toxicities, depending on the therapeutics, and immunophenotyping. So previously, I've talked to you all about or published about things like OKT3, which gives us a very short, more of an acute cytokine release. T-General14-12 and anti-CD28 agonists, where we see the peak cytokines are closer to 72 to 96 hours on our platform. We get T cell and myeloid and monocyte specific cytokines. We see things like body weight loss, lethality, high infiltration of the immune cells into the lungs, and evidence of single cell necrosis of the liver. And once again, PBMC donor variability. For bispecifics, we've done things like CD19, EGFR, and BCMA CD3s. Bispecifics, we've done dose ranging for efficacy and cytokine induction. We just submitted a paper for that, focusing on CD19, CD3. Donor variability and specificity data, looking at efficacy, cytokine release, and T-cell activation. One thing that's really quite cool, we're starting to move into a dual engraftment of tumors, both IV and memory fat pad. I'll talk more about that later on. But the idea is it captures short-term and long-term efficacy, multi-dosing studies, things like that. And we've demonstrated that this platform, and I presented this at HESI, I think 6 months ago, maybe end of last year instead, if the platform is reproducible. So let's get, we just submitted we published an abstract at ASCO with Bart Jensen's laboratory at Pfizer in a collaboration where we looked at essentially 4-1BB agonists, and we were able to show that the data we generated with this mouse model demonstrated that the model is clinically relevant for this class of antibodies, and we're able to reproduce what happened in the clinic in our mouse platform. We ran urelumab versus utomilab. We showed that the data is clinically relevant from the platform. The data is reproducible. We saw interferon gamma and myeloid and myelocyte-specific cytokines being induced. We saw severe liver necrosis with urelumab. We saw systemic toxicity, donor variability, and dose ranging as seen in the clinic. And I can present that at a later date, possibly. All right, so let's get into CAR T. That's why everybody's on the call. We are looking at CRS efficacy and expansion. We're looking at T-cell, myeloid, and myelocyte-specific cytokines being induced. We're looking at target and tumor-dependent readouts, and I'm going to present today is on liquid tumors, but we're moving now into solid tumor CAR Ts as well. I should also tell you that we are now in collaboration. We've been in collaboration with a clinical group looking at retrospective analysis of patient samples where we get the patient's PBMCs and the patient's CAR T and trying to see how our platform performs and be able to retrospectively show the same data in the mice. And that studies are ongoing and looks very promising according to the physicians. Okay, so CD19 CAR T. Everything I'm talking to you about is a PBMC platform. Okay, so just make sure we're clear about that. All the cytokines I'm showing you are human cytokines. Okay, so we got into this, and we know there's people out there now doing non-humanized NSGs where they put a tumor in, they put their CAR T in. Also, there's some nice studies being done using the CD34 humanized mouse models like the SGM3. The Nature Medicine paper came out like 3 years ago, showing some nice work. The feedback I'm getting from physicians from multiple hospitals is that these platforms are nice, but doesn't always mimic what you see in the clinic. And there also takes a little bit of time to develop these platforms. They can be costly, especially in the case of the CD34. So we wanted to develop a platform that was going to actually be more focused on not only autologous, but also the off-the-shelf or universal CAR Ts, where you're looking at the same CAR T being presented to an array of patients. And if we can be able to help them be able to find out how patients would respond to the therapy, because there's obviously going to be donor variability, patient donor variability, it would be a useful platform. So that was our focus. Our goal is to get the assay down to around 14 days. The platform usually takes about 16 at this time from beginning to end. So it's still relatively fast, close to 2 weeks. We want to look at individual responses to therapy, okay? And also we want to do efficacy and cytokine release and toxicity together. All the data I'm presenting to you is CRS. I can tell you that we're also doing neurotoxicity and cardiotoxicity with this platform as well. All right, so efficacy, CRS, and expansion of autologous CAR T in a PBMC humanized mice time course study. Let's do a time check. Okay, I'm good. Okay, we're using the MHC class 1, class 2 knockout, which has a delayed GVHD onset. I'm looking at PBMC donor 9534. I'm using 15 million PBMCs per mouse. In some mouse strains that I use, especially for clinical samples, I can go as low as 1 million PBMCs per mouse and get humanization. But anyway, in this double knockout, I'm using 15 million. And for the CAR T, which again was made from the same donor, I use 5 million CAR T. At day 0, we irradiate the mouse and we inject in IV our PBMCs. This allows us to get very good humanization of our mice. At day 6, okay, post PBMCs, we add our CAR T or our Mark CAR T or our PBS controls. 2 days later is the peak cytokine induction in our platform. So day 8, not all cytokines peak at day 8. Some peak at a later time point. I'll show a little bit of that data. And then we took the mice down at day 13. Now, in this experiment, I'm looking at PBMC humanized mice with no tumor. Obviously, B cells are CD19 positive. These CAR Ts are CD19 focused. I'm going to show you later on the addition of tumor cells to the platform. So just be patient. You'll see that as well. So right now we're just targeting the B cells. This is showing efficacy. This is CD19 numbers, okay, in the CD45 population. This is actual numbers per microliter. And then you're looking at your pre-numbers, okay. About 5% of the cells are CD19 positive. At day 2, okay, your PBS is in black and your gray is your CAR T treatment. You're expanding those CD19s up to around 150 cells per microliter, compared to percentage part of the CD45s. And you see no efficacy at day 2. Efficacy kicks in around day 6 and day 7. As you can see, all of the B cells are wiped out with the CAR T. Hopefully you can see my pointer. So you're getting efficacy about 7 days post-CAR T treatment. Cytokine. I'm having a problem with my mouse. You're seeing, you're looking at interferon gamma. Your cytokine release, like I said, interferon gamma, IL-10, IL-4. You're seeing a nice delta where you're seeing about 2,500 picograms per mil of interferon gamma. Your IL-10 is about 750, okay, and IL-4 is also elevated. I'm going to be bouncing around showing different cytokines at different times. We use a Luminex panel for a lot of our studies. CAR T expansion. You're looking at essentially CD3 positive cells per microliter of versus CD45s. You're seeing essentially no expansion at day 2. It's day 7, you start seeing the delta occurring, going from about 1,000 to about 6,000 with the CAR T. So this is one way of looking at expansion. I'm going to show you other ways in the next presentation, next experiment. With a different donor, 9531, we did the same kind of experiment. Once again, the protocol is similar to the last one we just did. Here's your autologous CAR T efficacy. It's in 6 days post-CAR. Essentially, the CAR is totally wiped out the CD19 population, going from about 750 cells down to per microliter down to essentially 0. We're getting good efficacy. Your cytokine release, interferon gamma, IL-10, IL-5, all coming up on 2 days post. Expansion, 3 different ways of looking at it. This CAR was actually flag tagged. So you're seeing day 6, a nice expansion. Essentially at 2 days, you didn't see much of it at all. So you're seeing a nice expansion at day 6 by either flag, CD45 levels, or your CD3s. Donor variability. This is a fun experiment. We used 9531 or 9534. these PBMC donors, and we essentially modified them to CAR T using the exact same construct. So essentially, we're just putting the same construct into two different donors, and we're comparing efficacy, cytokine release, and also any toxicity, and then also any expansion, I believe. So let me walk you through it carefully. Sort of left-hand side is body weight, and the right-hand side is your efficacy. Right-hand side is a little bit easy. You're seeing some differences in humanization between the different donors, but you're seeing complete response when it comes to efficacy with both donors at 7 days post. Essentially, all the CD19 cells are wiped out. If you look at body weight, which is the left-hand side, the CAR T was added at day 6. You're seeing a decrease in body weight is higher with 9534 than it is for 9531. And that's shown here by that dark purple line. So we're seeing efficacy with both donors, but we're seeing a hint of body weight loss correlating with one donor, but not the other one. You look at cytokine induction with both donors. You're seeing good levels of interferon gamma, about 2,500 to 3,000 picograms per mil. Your IL-10 is about the same in both, around 1,500. Your IL-5 is higher in 9534, which we saw some toxicity, at least by body weight. GM-CSF and MIP-1 beta. The expansion was a lot better with 9534, considerably better. And that's evident here. So we saw body weight loss with 9534. And we wanted to know if we added more target for the CAR T, will we be able to affect the body weight or toxicity possibly of this CAR T with this donor. So before we're using 15 million PBMCs, we just boosted it up to 17 million and repeated the study. The idea is adding more target because B cells are positive for CD19. Would that increase your toxicity? Here's the survival curve showing that's the case. Reason, which again, it's only 5 mice per group. So you're showing that 4 of the 5 mice in the CAR T treated group are dead by day 12. You've lost 1 mouse in the PBS control group, okay, which is probably due to the onset of GVHD starting to kick in. When you irradiate the mice, GVHD comes on faster. Okay, so you have to modify your experiments by using different cell numbers, different mouse strains, things like that. But we address that regularly in our platform. But you're seeing more target, increased toxicity with the same donor. Cytokine inductions, we have a time course here. PBS is in blue, CAR T is in pink or red. You can see we're getting a good onset of interferon gamma at day 8, which is 2 days post-CAR. Same thing with IL-10. Your MIG, your MIP-1 alpha, and your RANTES is coming on at day 11, which is essentially 5 days post-CAR. So we're seeing with a lot of stuff we're working with clinical samples, different sample onset of different cytokines does come up differently depending on the cytokines. Okay, so now I'm going to start talking about adding luciferase labeled Raji cells to the platform. So this allows us to do more extensive analysis of efficacy over a longer period of time without us having to require us to bleed the mice too often. It also allows us to start positioning the platform to understand how we would address solid tumors. And we can talk more about that at the end if you like, but the idea is to try to understand timelines, cell numbers, things like that. So in this experiment, okay, we have to again use an MHC class 1, class 2 knockout. We got 15 million PBMCs, we got our 5 million CARs, and we're adding a 250,000 Raji luciferase cells. Now, day 0, we're doing a little bit differently. Day 0, we're loading up our Raji cells first. The tumor is going in first. 6 days later, we're at our PBMCs. It gives us essentially a tumor a chance to expand. At day 6 is PBMCs. 2 days later, we add our CAR, letting the PBMCs to expand. And then 2 days later, we start assessing cytokines. At day 17, we terminate the study. Okay, so some interesting things came out of the experiment. So we did the experiment looking at with and without Raji cells just to see on efficacy. On the far left, looking at CD19 cells alone, you're seeing essentially good efficacy with the CAR T. All the CD19 cells are gone. With the presence of Raji cells, looking at CD19, we saw a drop of the CD19 positive B cells, but there's still some there with the Rajis there. Here's your luciferase labeled Raji expansion over a 9-day period. Okay, and the CAR T was able to eliminate all the Raji cells in the peripheral blood. Cytokine induction was a lot higher in the presence of the Raji cells. That was pretty clear. Interferon gamma without Raji in the far left, you've seen about 150 picograms per mil with this experiment. But in the presence of Raji cells, it's closer to 6,000 picograms per mil. Your IL-5 is a lot higher, about 10-fold higher in the presence of the Raji cells. So the presence of the tumor cells and or more target, you see an increase in cytokines. Your IL-3 and your GM-CSF is pretty pronounced as well. Raji cells are present, you're seeing significantly higher levels of GM-CSF. This is CAR T expansion. Once again, the expansion is a lot higher in the presence of the Raji cells. Without Raji cells, you're seeing about 225 cells, but this is flag labeled, versus about almost 8,000, okay, in the presence of Raji. This experiment was, of course, done at the same time, just in parallel mice. Comparison of autologous versus allogeneic CAR Ts. Now, in my laboratory, or the Jackson laboratory, we don't do drug discovery. All the tools I use, whether it's antibody or a bispecific or a CAR T, either I get from collaborations or I get it from, we purchase somebody that makes it for us. This, I talk about allogeneic CAR T, this is truly an allogeneic CAR T. It's not a universal CAR T, but it's able to demonstrate what we're trying to accomplish when it comes to the platform development. I'll show you why. So in this experiment, okay, we're looking at essentially, once again, double knockout mouse. We have 3 different PBMC donors, 9534, donor A and donor B. All 3 are 15 million cells per mouse. And I'm using one CAR T, 9534. So obviously we have an autologous PBMC with that donor, with that CAR T, and then we have 2 allo donors to the CAR T. The platform is shown here. It's a 13-day experiment from beginning to end. We irradiate at our PBMCs. We're targeting CD19, obviously, B cells. We look at day 6, we add our CAR T, okay, or PBS, and 2 days later we look at our cytokine induction and continue to day 13. So let's first look at the efficacy, and I'm going to go all the way over to day 7, okay, the very end. First of all, you should see is that in the black, you'll see that is your PBS control with the 9534 PBMCs. And you can see next to it in gray, we have complete response with the CAR T. The other two donors, okay, obviously are not, they're allo to the CAR T itself. We got less B cells by day 7, even though they were the same at the pre. You didn't get the expansion of the B cells with PBS like you did with the 9534. And we're also seeing great efficacy in the allo CAR T against those PBMC donors. So whether it's autologous or allogeneic, we're seeing really good efficacy with the 9534 CAR T. But expansion's a totally different story. And this is not going to surprise anybody doing universal CAR T work. The autologous, get the box around it at day 7, you've seen a really nice increase in the CAR T expansion by day 7. But you also look at the y-axis, essentially is broken, and just make sure you see that. And on the right-hand side is the expansion with the CAR T with the other two donors, and clearly we have very little expansion going on. So we have the allo response of the PBMCs against a CAR T, causing a less lack of expansion, which is similar we see with universal CAR Ts with issues with expansion versus the autologous. Once again, expansion comparison of autologous and allogeneic CAR Ts. One thing I've actually been surprised about this platform is that people haven't really embraced it yet for helping to pick or select a healthy donor to make the off-the-shelf CAR T. Because this assay allows you to actually take a healthy donor, run various positive controls for cytokine induction to look at the sensitivity of each individual to the induction of cytokine release for potential use for picking for donors for making the universal CAR T. But anyway, in this experiment, we're using a series of donors, and we're using 15 million PBMCs, and we're adding our 9534, same timelines. We're starting to ask questions about how individuals responded. So we have 9534 in purple CAR T, and we have 9531 CAR T. So both CAR T is going to be put into the same 6 donors. You got donor A, B, C, and D. You have donor 9531, which is a match to the 9531 CAR T. And you have 9534 PBMC donor that's a match to the 9534 CAR T. The cell populations in the vial, okay, are shown here on the right-hand side. So for each CAR T, there's 1 autologous match, and there's 5 allo matches, allo donors. And this is complicated. I'm going to try to walk through it slowly. PBS in gray. Your CAR T 9531 is yellow or orange. 9534 CAR T is in purple. Okay, and you see your donors on the bottom: 0595, 0901, 7691, 8595, 9531, and 9534. So let's work on the right-hand side. This is looking at efficacy. On the right-hand side, 9534, you're getting a complete response with its own CAR T. You're getting essentially all the CD19 cells are gone. With 9531 right next to it, so 9531 CAR T with the 9534 donor, you're not getting complete response. You're getting about, let's say, 40% of the cells, B cells are being eliminated. So you're seeing a difference in efficacy with the 2 different CARs for donor 9534's B cells. Next to it is 9531. We have the opposite effect. its own CAR T gives a complete response, while there's still residual B cells present with the 9534. Looking at the other donors, 8595, in this case, 9531's CAR is efficacious and the 9534 isn't. Same for 7691. There isn't much difference between 0901 between the both CARs, but 0595 is also more efficacious. Okay, sorry, for 0595, the 9531 CAR T is more efficacious. So what we're seeing here is differential responses with the different allo CAR T PBMC mix. In some cases with 9534 or 9531, it's pretty clear the autologous CAR works great for efficacy. In the case of a mismatched, okay, 9531 is the preferential CAR T being used against these donors versus 9534. Cytokine induction. Once again, you're seeing differential cytokine induction with the different CARs. We're not seeing any difference really between cytokine induction, whether it's autologous or allogeneic, which kind of makes sense. But we're seeing differential responses with interferon gamma, your IL-10 and your IL-5. In the case of interferon gamma, you're looking at anywhere from 6,000 picograms per mil or lower. IL-10, we're seeing a spike for 4,000 picograms per mil with donor 0595 with the 9531 CAR T. And then IL-5 also we're seeing pretty high levels being induced as well. So for cytokine induction, it's not clear autologous versus allogeneic whether it makes a difference or not. But there is a difference when it comes to efficacy, as I just showed you. A little bit of toxicity for 9534. We see the toxicity we saw before, where it's the autologous 9534 donor with the 9534 CAR T. We're seeing body weight loss. We're not seeing a lot of significant toxicity with body weight, except maybe in the case of the 0595 with the CAR T 9531, the orange on the far left. And I'm sorry if this seems a little fast. I just want to make sure we can get the data and give a chance for questions, and hopefully the presentation is being clear. Expansion's obvious. We don't get expansion if this is an autologous system. Look at day 8. And you can see we get really good expansion of the 9534 CAR T in the 9534 background, and very little expansion with 9531, but still better than any of the other cases. So expansion, if it's mismatched, you're going to have an allo response to the PBMCs against the CAR T, and that's pretty evident across these donors. Let's look at tumor burden, mouse body weight changes, and cytokine release. This is with Raji cells added to it, and this is just trying to help you define the platform a little bit better. Using luciferase labeled Raji cells and PBMCs. Far left, you're looking at the tumor burden increase, that's in blue and black. Okay, and then you treat it with either 1 million, 3 million, or 5 million CAR T, and you get a really nice dose response in this experiment. Whereas 1 million is the pink, 3 million is the red, and the 5 million is the dark red, showing good efficacy. The body weight loss was due to tumor burden. We saw that the mice are losing close to 20% of the body weight by day 18 without treatment of CAR T. While the CAR T treated mice, we didn't lose any body weight. Obviously, the tumor has been knocked down, so they don't have tumor burden loss of body weight. You see a nice little dose response when it comes to interferon gamma levels from the, once again, the flesh color or pink going up to the red to the dark red. Once again, we want to look at the high tumor burden and different cytokine induction. And essentially what we did here, let me go backwards. We did here is a little bit different. We actually added our Raji cells at day 0, but we waited 12 days to let the Raji cells to expand and take increased tumor burden. At day 12, we added our CAR T and our PBMCs together. And then 2 days later, we did a cytokine release analysis as well as day 15, and then cardiac bleed at day 17. And essentially what we're seeing here is really nice levels of interferon gamma at 2 days and 3 days post-CAR T. Our IL-10 is significantly higher. We're seeing peaks around 10,000 picograms per mil. And our IL-6, which is IL-6 is relatively low, okay, with low tumor burden. We've seen low levels always present in all our experiments, but it wasn't until we start putting in our tumor cells and letting them basically expand significantly do we start seeing things closer to 750 to 800 picograms per mil of IL-6 downstream. So for us, we're seeing a correlation with IL-6 with tumor burden. And I just want to thank you for your time. We been assessing this platform for CAR T. It's a lot more work to do. Obviously, the tools we're using are tools that we are having made for us commercially. I'm sure that most of you have CAR Ts that are a lot better quality than the ones we're using, but I think the data is going to be similar, just higher levels of readouts. We were able to show different levels of toxicity for different donors with the same CAR T construct. We can show efficacy in CAR T expansion. We're showing the difference between autologous and allogeneic. And we are now using essentially patient CAR Ts in our studies to understand better, using better quality CAR Ts. We're seeing the same effects, but essentially higher toxicity and higher cytokine induction with better quality CARs. And then all the collaborators we have working with us at various levels. My laboratory, the CAR T work is being led by Jiwan Yang. Obviously, I've got collaborators at Pfizer, Bart Jansen, NIBSC, Sandrine. Curtis has been very helpful with Sanish in helping us have a reality check about our platform as we develop it. Lenny Schultz for the work done on humanized mice, and our collaborators at University of Massachusetts. And I think I am done sharing.